Dr. John Bennett from InternetMedicine.com and Oncopedia.tv. We're here with a second show of Oncopedia.tv. And today the topic is personalized oncology, and we're being led in the discussion by Marcos Andre Costa from Brazil. Matter of fact, all the guests are from Brazil, but they're in different parts of North America right now. So we'll just go around initially and introduce ourselves and just say a little bit about where we're at, where we're practicing, and what's our connection to oncology. We'll start with you, Marcos. Oop. Marcos.
I'm ready.
Okay. Can you just introduce yourself and just say a few words, and then we'll go to the other guests?
Okay. Thank you, John.
Okay.
My name is Marcos Andre Costa. I'm editor of Oncopedia, and this is a Facebook fan page that is. This Facebook fan page is designated to show some information regarding head and neck cancer, lung cancer, and brain tumors. And today I work in Beneficência Portuguesa Hospital here in São Paulo. I'm a clinical oncologist, and it's a pleasure to be here.
Okay, Marcos. Louise, you have to unmute yourself, Louise. Oh, hold on, Louise. Unmute yourself. I make the same mistake. There you go. Okay.
Thank you, John.
You're welcome.
Yeah. So my name is Louise Araújo, and I'm a medical oncologist and a postdoctoral researcher, and I'm working at the Ohio State University in Columbus, Ohio. It's a pleasure to join this group of friends tonight.
Very good, Louis. Welcome. And Philippe, the first Philippe #1. Philippe Battalini. Can you unmute yourself?
All right.
Okay,
yeah.
Go ahead.
Hello, I'm Philippe Battalini. I'm a physician here from Guyana. I just arrived from UCLA, where I was doing research, and I do work in a cancer hospital here at Guyana, basically taking care of all kinds of medical emergencies. So I usually stay as a night float. So yes, that's my connection with oncology, and I'm applying for internal medicine in the U.S. for later pursuing oncology residency.
Very good. Welcome, Philippe.
Thank you, John.
Philippe #2.
Hi, everyone. Thank you for the invitation. So my name is Philippe Ades. I'm a clinical oncologist. I stayed 5 years. I studied actually with Louise and Marcos Andre in the National Cancer Institute in Brazil, where we did our training in medical oncology. And I stayed 5 years in Europe doing research, and I acquired a master's and PhD degree in mostly breast cancer research. Now I'm back in Brazil. I'm working in São Paulo in clinical practice and also in research in the Albert Einstein Hospital.
Very good. Welcome, Philippe. Marcos, say you're the best, and it's good to have you here. Okay, I guess I'll turn it over to Marcos. He's going to coordinate the discussion. So it's all yours, Marcos.
Okay. Yes, I told you that Philippe and Louise, we have a great panel today. These guys, they are very skilled guys, and it's a big pleasure to me to be part of this panel. They are very differentiated guys, really. And I will start this Oncopedia TV show talking to Philippe Ades, and I would like to add something to Philippe. Philippe, tell us something about your research there in Durable the hospital, and tell us the line of research that you conducted in the last 5 years there, please.
Okay, thank you. Actually, I stayed 2 years in the Gustave Roussy in Paris, where I did mostly translational research in melanoma. So at the time, we had the initiation of the BRAF inhibitors and actually also the anti-CTLA-4. So it was a very exciting moment for melanoma. It was the first time in the history of oncology where we had a medication that really changed the outcome of this patient. So what I did there was most translational research looking for mechanism of resistance to target therapies and we try to establish a molecular signature for sensitivity to these agents, but unfortunately we were not very successful at the time with this kind of approach. So in phase 3 trials actually, right? So I had a job that was a medical advisor for these big phase 3 studies, mostly the HERA, the auto study, and actually the ongoing affinity study, where I spent most of my time looking for the adjuvant pertuzumab. And parallel to this, I developed my PhD project that was in public research, so how we could apply the knowledge that we acquired in clinical and translational research to clinical practice. So how can we improve access to new medications in real-life situations?
Okay.
And great, Philippe. And we told behalf inhibitors and pertuzumab there is a HER2 inhibitor. This is part of the theme of this hangout, that is personalized oncology. And talk to us in a few words, in a few minutes, the main action of this modern inhibitors, and what is behalf, what is HER2, and what are the main mechanisms of action of this drugs that you studied there.
Exactly. So in breast cancer, that was most of my recent focus in research. The field of research changed a lot since the 2000s. So we have a publication in Nature, or Nature actually, from the Stanford group, where they really identified 4 main intrinsic subtypes of breast cancer. So from this study, we start to understand breast cancer not as a single disease, but different diseases that arise from the same part of the body. And then we have to develop different research strategies target to every one of these diseases. So the HER2 subtype of breast cancer is around it has a prevalence of 15 to 20% of the patients. So it's characterized by the overexpression of this protein, and then this protein drives the oncogenic mechanism. So once we develop monoclonal antibodies and inhibitors of tyrosine kinase to this target, we improved a lot the outcome of this patient. So the biggest drugs in the market now that target the HER2 receptor, the most effective, most promising are the established already is trastuzumab, is a monoclonal antibody that really blocks this signaling and actually flags to the immune system to destroy the cancer cell. And we also have pertuzumab that has a similar activity, but to another receptor of the family, that HER3 receptor. And we have lapatinib now approved. Then is a tyrosine kinase inhibitor that blocks the signal from inside the membrane, but it's not as effective as the 2 previous molecules. And we have more things going on targeting the same pathway. On the other hand, we talk about the BRAF. So this is mostly important in melanoma. This is it's a normal protein, but has an oncogenic mutation. So when it's mutated, it overexpresses a signaling pathway that tells the cell to multiply, to grow, and to invade. So around 50% of the melanomas, they express this mutation. And when you block this, not only with the BRAF mutation, but now with the MEK inhibitors as well, we have dramatic responses in melanoma that bears these mutations. So now we identify that different diseases have different mutations, and targeting these specific abnormalities improves a lot the outcome of these patients.
Great, excellent, Philippe. And we know that lung cancer have HER2 and BRAF mutation, Louise. This is part of your main research there in Ohio today, or not?
Yes, it is. So actually, BRAF is present in about 5% of lung adenocarcinomas, and it's also present in a little bit of squamous cell carcinomas of the lung. And And we are learning now that BRAF can be mutated not only in melanoma, but also in lung cancer, but also in other diseases. And one thing that we found recently is that BRAF is not the only protein in the RAF family that can be mutated. So we had a patient who was enrolled in a clinical trial with sorafenib, and as you guys know, sorafenib is a broad tyrosine kinase inhibitor. It became very famous in renal cell carcinoma because it blocks VEGFR and PDGFR, so it blocks angiogenesis, which is a key factor in kidney cancer. But sorafenib is very broad. Sorafenib blocks, it was actually developed as a CRAF inhibitor. And CRAF is one of the proteins in the BRAF family. So you have ARAF, you have BRAF, you have CRAF. You have three different proteins in mammals. And sorafenib was developed as a CRAF inhibitor. So in this clinical trial with about 200 patients treated with sorafenib, we had only 9 responders, only 9 people whose tumor shrank. And out of the 9 patients, we had a single patient who was a magic response. So this female, she was about 59 years old, had a lung cancer, lung adenocarcinoma. She was a former smoker, and the disease was progressing over all the chemotherapies, including TKI. She also had erlotinib, and the tumor was resistant to erlotinib. So we did a whole genome sequencing and whole transcriptome sequencing, and that's what we call the outlier responder analysis. And we found a mutation in ARAF. It's a mutation in the position called S214. So it's a S214C mutation. And actually, this mutation was in a negative regulatory domain. When you have this mutation in the negative regulatory domain, you open the protein, so ARAF becomes open and potentially active. And by being opened, ARAF can dimerize with CRAF and activate the downstream pathway, which goes to MEK. So we showed in the lab, coming back to the lab, we showed that this mutation in ARAF can actually be a driver in lung cells. And we're now going to prove that in mouse. So that's just one example that BRAF is not the only mutation that can happen in the pathway. You can have different mutations in different diseases.
Great.
Marcus, can you give me 1 second?
Sorry, Luis.
Okay. Okay, I would just like to add one thing that is very interesting, and not always one thing that we think is logical. It works because the disease setting is also very important. So when we developed the BRAF inhibitor, at the time was called PLX4032, was the vemurafenib. So he's a very potent inhibitor of the V600E RAF protein, right? So people started looking for this protein mutated in other diseases. And then we realized that in colon cancer, it was mutated. We have a percentage of colon cancers that have these specific mutations. So the logic thing is, let's block this protein in colon cancer as well. And when we did this stuff, it doesn't work. So it's not only the mutation, but the context where the mutation happens is very important for the results of the drug. So we are starting to learn a lot about the cell mechanisms and the mutations and the drivers of cancer. But we still see that there are many things that we don't quite understand. Epigenetics coming in, editing things. There's lots of new molecular biology that we need to learn to really improve the outcomes.
Excellent. This is very important, Philippe. It's very important. The epigenetic mechanisms that are involved in the microenvironment of tumors and the function of the genes and the sites of mutations. This is very important because no matters is the mutation is present, but the microenvironment, there are very mechanisms and other factors that are involved to make that mutation to work and become a driver on oncogene. Absolutely right.
This example that Louise gave us now, the one single specification that has a, I think we're going to see this more and more in the future. I think we understand that there's no subgroup of disease, but maybe we have single diseases, right? Everyone has one specific disease, and I think the great challenge to the future is to establish clinical trials and research strategies that can really target this N1 trial that we say. It's a trial that tests this specific drug for these specific patients. So how we're going to develop these strategies to move forward with research. I think we have some interesting already initiatives going on, but we still have a lot to do. So it's a very exciting moment for research.
Can I add something here, Marcus and Philippe?
Of course. Go ahead.
I'm sorry I had to be absent. So trying to add to what Philippe was saying and what I said before, we now have this label, personalized medicine, right? And some institutions are naming personalized medicine as precision medicine, targeted therapy medicine, whatever. But and there's also different infrastructures to implement personalized medicine. So one idea is the very early diagnosis. That's, for example, BRAF in melanoma, and for example, trastuzumab and HER2 in breast cancer. And I'll include here EGFR and ALK in lung cancer. So those are established personalized medicine. If you have melanoma nowadays, before any decision on the clinical setting, you're going to test for BRAF. And we are talking about metastatic disease. If somebody is watching we're talking about metastatic disease. We can talk about adjuvant later, but if you have breast cancer, there's no single person in this world who is not going to test for hormone receptors and HER2. Lung adenocarcinomas, everybody's testing nowadays for EGFR and ALK. Some people test for KRAS, but KRAS nowadays doesn't change therapy. So that's frontline. That's frontline. The second strategy is the patient who progressed over chemotherapies, over the standard of care. So that's the melanoma patient who doesn't have a driver and is progressing under chemo. Is the lung cancer patient who doesn't have a driver, has taken 2-3 chemotherapies, and has no further option or a standard of care option, or the breast cancer patient who has progressed, if that's possible, not to have a line in breast cancer, but let's say it's the third line, fourth line, and you don't have a target. So that's a different personalized medicine, but it's also personalized medicine. And that's where we try to implement this kind of very wide tests. So one of the examples is a company in the US called Foundation Medicine, but different institutions are applying their own tests. So it works like clinical trials. You usually have a consent form just to test the tumor. And by testing the tumor, you try to assess different genes, usually more than 100, 200, 300 genes. And then you try to find a new mutation. It can be a melanoma mutation in a sarcoma patient. So it could be a BRAF mutation in sarcoma. It could be a FGFR fusion gene in a sarcoma. It could be a EGFR mutation in a different cancer. And then the idea is the initial idea is to put these patients in phase 1 clinical trials, or phase 2 trials, but the big idea to test these patients came in a strategy to enrich phase 1 clinical trials. So you're not going to give a drug for somebody who has a very low chance of response. We all know the chances of getting a response in phase 1 clinical trials are very low, very low. But we expect that if we enrich patients, if we get all the FGFR fusions, mutations, overexpression, amplification in a single group for an FGFR inhibitor, we expect that we're going to find better responses, and we expect that we're going to approve these drugs faster. And that's one of the strategies. The third strategy is exactly the one I was saying before. It's like the outlier responder. So this is a patient who you didn't expect very much to respond, or a patient that you expect a lot to respond. That's a HER2 amplified breast cancer who doesn't respond to lapatinib or to one of the HER2 drugs. And if he doesn't respond, there's something else in the biology that should be able to explain. And that's what we call outlier responder protocols. And then we want to sequence what, if you can afford, you want to sequence the whole genome and whole transcriptome. If you cannot afford, you go to smaller panels, but still, what you're looking for mechanisms of resistance. Even though they overexpress HER2, there's something else that's going to explain biologically why this tumor is not shrinking under HER2 blockage.
And this is a very strong trend that just in phase 1 to gather these patients in phase 1 and enrich the phase 1 trial with patients point mutations, and if it was what happened with ALK, the big data in the initial data started in the phase 1 trial that some years ago, and of crizotinib, and based in one phase 1 trial, this drug crizotinib is an ALK inhibitor. He became available to use in the clinical practice, and he received a breakthrough of FDA. It's a very strong trend today in oncology. Would you like to comment, Philippe Ades and Philippe Battalini, about what Louise told for us?
You want to go ahead, Philippe, or can I just catch the line here? Guys, so I think you touched very interesting points, right? The thing that every patient has different mutations, and tumors, they have mutations that we don't really expect. I think there's something here that we have to, as a scientific society, to really think about it. But because now, what we used to, I'm going to push to my my drug approval and the real life situations. What we used to approve drugs, to make them affordable, to make them then go to the market, is phase 3 studies. So we have to test the drugs against the best drug that exists. So we see that this population, because we think in the past, we used to think that the diseases were similarly equal, right? Even when we talk about HER2 and estrogen receptor, it's not really I agree that it's the beginning of personalization, but now, as we get more knowledge about this stuff, we see that it's more like stratification and not really personalized therapy, because we have, like we said, we have a HER2 mutation in one specific tumor, but in another mutation that can join together and drive the oncogenic processes. So I think that as a society, and we have to join with the health authorities, and maybe it's a time to share risks, right? We have to really explain to the patients until where we exactly know what's going to happen, and then maybe blockage of the another pathway with this foundation one, for instance, approach can work and cannot work, but I think it's an interesting approach. So I think if we share these doubts and risks with the patients and with the health authorities, and not only give the drug, but we give the drug and we collect information so we can have a public database, we can really be building knowledge over time, right? Because this patient, they will be unique. We will never have a phase 3 trials that can test one patient with 3 mutations against other patient with 4 mutations with one specific inhibitor. These things are very good to I'd say, bring questions into clinical research, to be very provocative, but in real life situations we have to deal with doubt, and then maybe we have to share this doubt with the patients and the health authorities. They have to understand that we're moving into a moment in oncology that we will not have this 100% certainty that we used to have in phase 3 trials, right? Even because we see in phase 3 trials that response rate 40%. So 60% of these people, they don't respond, right? But if in one group 40% respond and in the other 10%, we approve the drug. So I really think we have to change our research, but and maybe we have to accept. It's very difficult for us as doctors to accept doubt and pass this doubt that we have to the patient, but it will be a shared treatment option, right? I think people have to be more informed and to accept a bit these things.
Can I add here, Marcus? So just coming back to the example of the ARAF. So ARAF is mutated. We checked it back on TCGA dataset. TCGA has a little more than I think 200 or 300 lung adenocarcinomas. So we went back to the dataset and we found that ARAF is mutated in 0.5% of cases. The same mutation, 0.5%. And we looked at a similar mutation in CRAF. So if you look at the very homologous hotspot in CRAF, there's also mutations in the same. It's S214 in ARAF and S257 and S259 in CRAF. So if you account for this combination of the mutation in the same domain, it's actually 1% of lung adenos. We went to the other data sets in TCGA, for example, gastric cancer, something. And if you look at this big number, it's thousands of cancers now, it's about 1% of cancers. So it's not only lung cancer. So it's a very rare mutation, very rare mutation. First thing, you can't find rare mutations in the kind of studies that we have nowadays. Because for example, TCGA lung adenocarcinoma has a power. You need to filter a lot of stuff because you find a lot of passenger mutations that have no activity. So you filter a lot. And the power of detection is in the range of 5 to 10% of frequency. So TCGA right now, which is it's like the Human Genome Project, it is a cancer genome project. It doesn't have enough power to detect a mutation that has a frequency of 1% in the population. It's going to be discarded. So one thing is we need thousands of patients sequenced to figure out rare mutations. And that's why TCGA is not enough. We are keep going with the international collaborative genomic projects in cancer to get these higher numbers and find new mutations. The second thing is with physicians, medical oncologists ordering this kind of tests nowadays we have a lot of clinical information coming back. Every day we have oncologists ordering this kind of tests. So we have a lot of clinical information. And we don't have the same high throughput technology to prove if these mutations are important or not, and which drugs are going to respond to this kind of mutations. So we are in a higher throughput of giving the drug and testing, then we are in the situation of proving. So for example, one of these tests, let's say you have this your lung cancer patient in your clinic, and you say, okay, he responded to nothing, he has no EGFR mutation, he has no ALK translocation, I've tested for ROS1, RET, nothing, no oncogenic driver. And you say, all right, I'll test him with the fine comprehensive test. And you find a mutation that has never been reported before. I mean, you have no idea if he's going to respond or not just because it's a mutation in let's say in PI3 kinase, and you have a PI3 kinase inhibitor, a PI3K inhibitor. It doesn't prove that this patient is going to respond, right? So it should be tested in a lab setting, in a preclinical setting, ideally to have a better clue if this patient is going to respond. Problem number 2, cancer is very complex. We're not finding cancers like GIST, the gastrointestinal stromal tumor anymore, where you have a single alteration or a very classic mutation, very low number of mutations, and you give imatinib because of a mutation in KIT, K-A-I-T, you give a KIT inhibitor with imatinib, the tumor shrinks, and the disease is under control for years. We are not finding CML anymore, single translocation, and you give imatinib. Basically, you have decades of control, decades. So now we're finding lung cancer patients with a mutation that's probably a driver. Lung cancer mutation has about 10 mutations per mega base pairs. So it's at least 100 times higher than CML. So you have not only a driver mutation, but you have a lot of noise. And this noise can be just passenger, or they could be somehow networking with the driver mutation that we're calling, and it could impact the response to therapy. And there's no high throughput technology. There are some high throughput technology, but not ideal high throughput technology to test if those 100 mutations that we found in patient 1, can we test if giving a cell 100 mutations and giving the drug, which drug is going to respond? We don't have this kind of technology nowadays. So in the clinic, in the very clinic, when you're going to see your patient, order the test, get a list of most important mutations, and you share your idea that it could respond to that drug, but we have to understand we're in a very preliminary step in drug development, and we're moving drug development and moving phase 1 and moving testing very fast to the clinic. So we really don't know what's going to have a response, a tumor shrinkage, or nothing. So I'd like to hear from Marcos, Philippe, if how is that the testing situation going on in your practice nowadays? How have you been moving to this personalized in the practice nowadays?
Philippe, go ahead, Philippe.
According to what I have to say, totally goes along with what Philippe said before, and as we keep finding outliers, it sounds to me that it's very, very hard, or maybe nearly impossible, to figure out proper treatments and to assess outcomes in patients if we do not work better on this international database. It's kind of scary when we stop talking about mutation and kind of start discussing about epigenetics, because the number of variables that we need to handle when analyzing data, it's just too much. So what I really think is that there's no other way to go further in this kind of research, and without working in a collaborative way with a lot of research centers throughout the world. I mean, it's just too individualized. That would be very, very hard to prove scientifically with enough numbers to approve drugs to basically finally help the patients.
Can you just add to this? I think I agree completely with you, right? The way to move forward here is collaborative. And I think Louise, before mentioned the TCGA, TCGA is one of the amazing examples of cooperative work that we had recently in oncology. So we have labs from all over this planet, people putting samples from every kind of cancer you can imagine in multiple platforms, and now we can cross-check this information where they match, like put them mix and we have to go in the direction of really do this with clinical practice as well, right? When you have a situation that when you use a test like Foundation 1, we had recently a patient that had a COOP, like a tumor of unknown primary. So the results of Foundation 1 pointed to sensitivity to afatinib, navelbine, and Taxol. I would never do this combination. I don't think anyone would do this combination. She had a completely response through PET, and she was without any disease for 1 year. This is unknown primary. It's a very, very bad prognostic tumor, and she was in complete response for 1 year. So if we could put this database, this information in one specific database, and people could access, and it could be building this knowledge and actually sharing the doubt, because this was completely out of any protocol, out of any kind of guideline. It was exclusively by the results of the Foundation 1. So building this knowledge time over time is very interesting. And I think there's already initiatives. I think the FDA has one, and the EMA, the European counterpart of FDA, they have a process now that is called adaptive licensing, and then it's exactly like this. You test a drug in one specific set, and then you try to approve in that specific setting. And then people will use, and then as you go building knowledge, the indications, they go extending and extending until full approval of the drug. I think it's the only way we have to do this with the new drugs and the diseases that becomes very rare. If you put like a patient with a mutation in EGFR and ALK EMN4 translocation and maybe have a P53, how can you check for all these specific mutations and know that, okay, this patient with this 15 kind of mutations will respond to this specific drug? It's impossible to know this thing. It's just with the building of the knowledge, and we have to build this community that make, I would say, a clinical TCGA at some point in the future, and sharing the doubt as well, because we'll never be sure in life. I think this is the way we have to move forward, from the choice of the treatment to the drug approval, right?
Yeah. This is very important. We are seeing today a big effort to tailor the best treatment for the patient, and some groups, they have a very soon and they are very fast in sampling the patient and send to United States to Foundation 1 to see, for example, for lung cancer, metastatic lung cancer, they look for ALK EGFR mutation. There is a big effort some time to identify this mutation, but the next step is we will be able to use targeted therapy for these patients in the clinical practice. I think that no, the response is no. We can identify to mutation or alteration, MET, RED, anyone, but the access of the patient to these drugs, these new drugs, they are very. This is a big problem today. The health insurance, they do not cover these drugs, and the big question is what to do with that information. We know that the patient has that. The oncology has evolved a lot in toward this direction, but sometimes we do not know what to do with that information. It's a big concern, it's a big question today, and this is the reason, because of many of my colleagues do not send the tumor for further analysis But this is the last problem. But the problem, the beginning problem is many of clinical oncologists, they do not investigate EGFR, just the initial, the very beginning investigation that there is EGFR mutation or ALK translocation. Many of oncologists in some parts of Brazil is a very heterogeneous country, and we know that many oncologists, they do not investigate this very simple, this very important alteration, mutation, and translocation in the upfront of lung cancer, metastatic lung cancer patients. This is a very concerning problem. This is one problem. The second is our public health system that is not prepared to offer even the more basic treatment for the patient. You have some very severe restriction to some chemotherapies and radiotherapy. It's very heterogeneous here in Brazil, but it's become better and better and better along the time. I'd like to know of Philippe and Louise, if in Brussels, in UCLA and Ohio, what is the percentage of the oncologists that in case of breast cancer, for example, they investigate or two, or lung cancer, they investigate for EGFR and ALK? I don't think that in Brazil is very high.
Well, if I can get started, I believe the percentage to test for EGFR and ALK should be very high. And a lot of people like to order KRAS, which I don't think it makes a lot of sense nowadays. So if we go to NCCN, for example, in lung cancer, NCCN is going to recommend testing all samples with an adenocarcinoma component, and actually we are moving to every sample which is not a pure squamous cell carcinoma. We're testing all the samples to EGFR and ALK, but ideally, and NCCN accepts this as a possibility, ideally, if you can get more information, why not to get more information? And if you check the prices of doing a next-gen sequencing approach, where you're going to test for a lot of not only mutations but fusions, and you can include RET, ROS1, and EGFR, ALK, all these most important genes, MAC1, AKT1, MET amplification, MET mutations. Why not to get more information? The big thing is ROS1, for example. ROS1 is present in about 2% of lung adenocarcinomas, and ROS1 fusions are clearly associated with sensitivity to ALK inhibitors like crizotinib. RET, the same thing. RET is present. Fusions are present in about 2% of lung adenos, and I told last time in our last Oncopedia TV show. In the phase 1 clinical trial enriched by RET fusions, with the first 3 patients enrolled, 2 patients had a beautiful response. 2 patients out of 3, and that was the fastest publication, even faster than ALK discovery. So RET fusion genes are getting established, are becoming very established in lung adenocarcinomas. And you're not going to find patients with this kind of alterations every time, but if you're looking for drivers altogether, you should see at least 30 or 40% of samples. Some databases talk about 50 or more, but I think it's a very enriched database, like the Lung Cancer Mutation Consortium. They had a lot of never smokers, a lot of females, so I think they're enriched. but you should expect at least 30 or 40% of lung adenomas having a different driver. And these are not magical drivers. These are not difficult drugs to get access to, right? Crizotinib is there. Crizotinib is there. EGFR inhibitors are there. RET inhibitors. There's a bunch of clinical trials with RET inhibitors. And you have drugs approved for thyroid cancer, for example, which can be used in RET-fused lung adenocarcinomas. So NCCN recommends, in summary, NCCN recommends EGFR and ALK, but if you have access to the testing and it's affordable, it's acceptable to test more things, more stuff. And I'm clearly in favor of testing more, because you can not only understand better the biology, but you can have a chance to put this patient in a clinical trial or have some kind of compassionate access to one of these drugs. And the patient can really benefit if you find a driver. That's no question about that. Before you guys move on, I think we could come back to the immunology talk interacting with genomics, but I think Philippe may want to move on with the discussion.
So should I continue? Okay, so yeah, this is a very interesting thing. So we have lots of markers, and we're not really sure what to ask in clinical practice. So when I was in Brussels doing my PhD, we were interested about this topic of what people use in real life situations, right? What are the targets that really change your clinical practice? Because we see the Sangarian consensus that is one of the greatest consensus about breast cancer treatment that we have today. This is based on people's opinions, right? So, and of course, it's not people that regular people, they're really specialists in breast cancer in all over the planet. But anyway, we're interested to hear opinions of people in the field, right? So we sent a questionnaire with multiple areas of uncertainty in breast cancer. And one of them, a couple of these questions were regarding which are the targets that you really use in clinical practice. So most of the people, I think 100% of them, for breast cancer, they use estrogen receptor, progesterone receptor, and the HER2. These are the things that really change your clinical practice. And then you see after this have people asking Ki67, that is a marker of proliferation, with all the problems that Ki67 has, right? Because it's very difficult to standardize and to say which is a high proliferation and which is a low proliferation, but people still use it. And then you come with all the markers that really don't change clinical practice in breast cancer, like cytokeratins and other things that are minor. So people also use a couple of genetic tests, like oncotype, to tell if the disease has a higher risk of recurrence or not. Is a test that is being validated in a prospective trial, but it looks interesting. And we have a similar method that is called MammaPrint, but people use it a little less because it's a European test, and then also request some validation. So actually we have lots of things, but in clinical practice, really validated for breast cancer, we have these 3 targets, right? So that's why I think when we have different things, we have to compose this prospective database. In clinical research, you have more a different strategy. We have a very interesting initiative by the Breast International Group, and in collaboration with the colleagues in the United States. So they are doing a kind of umbrella group of tests, a group of studies. So in the beginning of the study, patients are tested for a lot of mutations, right? Very, very long list of possible targets. And then, according to the mutation, they are enrolled in specific trials, testing drugs, blocking these specific mutations. So we have this really one single big protocol that will enroll all the patients, but the drugs, they will be different depending on the target that the patient has. You have also the I-SPY study that is more or less like this in the United States. So I think we're going to be validating more targets in the future, right?
Great. I think that we can move on to immunotherapy. I will start with this, talk my recent experience there in Santiago, Chile, where I went to a research meeting with PD-L1 inhibitors, and we discussed for 2 days all the mechanisms of action and the rationale for this treatment in lung cancer, mainly in lung cancer. And we have a lot of data in melanoma. There is a very aggressive skin cancer and very impressive response. The beginning data, and Philippe can talk to us a little bit more regarding this, about this, the data in melanoma, but in lung cancer we have recent data. We have recent data in head and neck cancer and triple negative breast cancer, and more recently in Hodgkin lymphoma. This is a very fast moving area in oncology, and in the future hangout we can discuss more about this. But Philippe, tell for us something regarding immunotherapy and your experience in phase 1 trials and in clinical use of this kind of drugs, pembrolizumab, nivolumab, and other. And you Louise, the same.
So guys, this is a very interesting direction. So it's a big topic, this thing. I think it takes, if we're going to go into this topic, it will take one hour. But it's a very promising strategy. So they are inhibitors of checkpoint in immunity. We have these two really interesting drugs, pembrolizumab and nivolumab. Both of them were plenary sessions in the ESMO 2014 this year in melanoma. These things are very promising, but actually the strategy there is a bit different. I don't know quite exactly where you're going to, right? Because you are stimulating the immune system to attack your cancer cells. And at the other time, on the other hand, you are trying to kill your cancer cells with cytotoxics and target therapies that maybe they reduce the immune system activity. So how are we going to put them together? I'm still not really sure. I think in the future we're going to understand a bit better how it works. There are some people that think by killing the cell we expose the antigens, so this can boost the immune response. So there's also a possibility of how this drug works. And they work very well in immunogenic tumors, right? Tumors that have lots of targets. So melanoma is one of this kind of tumor. Lung cancer is also one of these tumors. And in breast cancer, we see the most immunogenic subtypes that are HER2 and triple negative breast cancers. And we have already data on this, preliminary data, like phase 1, phase 2, but showing that these drugs, they look promising in this kind of cancers as well. So it's a whole new field, very promising. I think in the future we'll see lots of stuff coming in.
If I can go ahead.
Okay, Louise.
I wanted to show how this, as Philippe pointed, how is personalized medicine and immunotherapy, is it different things? Is it the same stuff? I mean, the big thing about immunotherapy, it's so fantastic. I mean, we're seeing responses. The response rate is not so dramatic. In lung cancer, it's a 20% rate of responses, but it's very prolonged. So the 20% responders, some of them are alive and without disease for 3-4 years now. These people may be cured, which never happened in lung cancer, right? We don't have a treatment nowadays. We have seen some breast cancer survivors who are disease-free after 5 years. I've seen that before, but that's not the standard. That's not the standard. And with immunotherapy, it's very exciting. But we have no biomarkers for immunotherapy right now, right? And how is targeted therapy and the classic personalized medicine, which is genomic testing? We're looking for mutations. We're looking for signatures like the oncotype, DX, nanoprint, or we're looking at expression, immunohistochemistry for HER2, amplification, whatever. How is this information going to interact with immunotherapy? So I just wanted to point, first thing, yes, there's a fight. Who is going to win? For example, in melanoma, I've seen experts in melanoma pointing that right now, if they find a BRAF mutation in melanoma, some of the experts prefer to give immunotherapy first, even knowing that BRAF is the likely the driver and can have a dramatic response to vemurafenib, to MEK inhibitors, the brafenib, whatever. Some of them prefer to start with immunotherapy because the time of response is longer, in average, is longer than with the TKI, which is very funny. And the second thing is, how is these drugs going to work together? So, if you find a BRAF mutation in melanoma, can you give a BRAF inhibitor along with immunotherapy? Is it better? Is it worse? And the clinical trials are going on. In lung cancer, the same. You find an EGFR mutation, standard of care, TKI. And there's a whole approach to the TKI in the second line, third line. You may want to rebiopsy, resequence, and then decide what your options are. Third generation, second generation TKIs, or immunotherapy. How is combining them, these options going to work? The clinical trials are going on, so there's no answer. Adding chemo on top of immunotherapy is another big thing, as Philippe pointed. Maybe killing the cells, you're going to show up your antigens, and by exposing antigens to antigen-presenting cells, it might increase the immune response and might be better for the immunotherapy. We don't know. And the third thing, we need to find biomarkers for immunotherapy. As only about 20% of people are actually responding, maybe polymorphisms in lymphocytes could be responsible for the response, no response difference. Maybe can the response correlate with the specific mutations in cancer, like TP53, EGFR, KRAS? Is there any higher response rate according to the frequency of mutations? Is immunotherapy going to work better in BRAF mutated melanoma, in HER2 amplified breast cancer? These are all open questions. So the interaction between personalized medicine and immunotherapy is an open field. And that's going to be very exciting to deal with this kind of questions in the research setting and in the clinical setting in the years to come. I think it's a really exciting area.
Great. I think that the biomarker in immunotherapy it's a crucial point. Some preliminary data have shown that that tumors that has PD-L1 expression compared to that patient that has tumor that are PD-L1 negative, they have a higher response rate to pembrolizumab, for example. Keynote is a preliminary study, a recent study that showed that it was almost 61% of response rate for that patient that had PD-L1 expression compared to almost 4 or 5% of that patient that do not have a PD-L1 expression. This is very important. I think that some trials today, they are enriching the study just with patients with PD-L1 expression, and I think that this will be the future of these trials, to enrich this patient with this trial with this kind of patients. Philippe, could you comment something like this? Some data regarding this PD-L1 expression as a biomarker in immunotherapy.
Okay. So, can you hear me? Okay. So actually, we come back to a problem in biomarker research, that is what is predictive and what is prognostic, right? So when you see the PDL1 is a very interesting target. So it's a protein that is expressed by normal cells, but actually to the tumor that tells the immune system to stop the attack, right? So if I have the PDL1, it says to the immune system, stop it, don't kill me. So if you block this signal, the immune system go there and kill it, right? So this is what these drugs do. They stop the cancer from telling the immune system to stop killing it. Actually, in melanoma at least, it looks more like prognostic than predictive because of, as you said, if you have the protein, you respond better. But people that don't have the protein also respond to a lesser extent, right? But they still do respond. So I think it's not only this, but maybe another target around there that is always playing a role. So now I would not recommend using the expression of PDL1 for doing or not doing the drug. It's mostly a predictive information in my point of view today, the expression of PDL1.
John, any question, John? Philippe, Battaglini, any question? Luis?
I totally agree with Philippe.
Great.
PD1 is not the perfect biomarker yet. We have to keep looking for it.
Battaglini?
Actually, I'm just learning here. You guys rock. So, as a general practitioner, I mean, I'm just writing topics here just to read some research to get a little bit closer to this discussion. But it's just been great for me here to be here and participate. I'm just learning a lot. Thank you, guys.
Yeah, you know, Felipe, I'm in the same boat. I like the discussion. You know, one of the challenges. I'm just more or less a general practitioner, but I guess one of the challenges is putting all this data in an international database, which I know there's a lot of problems: privacy, politics, money, security. But I guess ideally, in the best of all worlds, we'd have one big database where people like Felipe, that case you mentioned, where they had a really good result. I guess we really won't benefit unless we know about that. All these things that are happening in different parts of the world with different combinations of therapy, we won't know about until there's a really good international database. What do you guys say about that?
I think that we're building the need for this now. I think that our medicine is evolving to a point that we have to have these things. If we don't have an international, or at least a national database, target to one specific health authority, and I think this will be driven mostly by the physicians and pharma companies pushing the government to approve drugs in specific situations. So then we have to create this tool where we have to compile data over time. So we learn from the fewer patients that have these very specific and punctual mutations and how to really individualize therapy. I think this is a continuous process. Is a learning curve that will be just going up. I think we have a.
Okay.
John,
Yeah.
the other interesting approach is not only feeding the database, but there's people also working as a matching database, just like people do with marriage. So the guy go to the website and put all his information, right? I'm a gym teacher and whatever. I live this place. I like dancing. I like music, and they match the guy with the perfect girl. So there's people working the same kind of thing for cancer treatment. Okay, I get my result, or the physician can also look up. I get my results. I have KRAS mutation, STK11 mutation, it's a lung cancer, it's an adenocarcinoma, it's didn't respond to chemotherapy, and then they match him in this data set. Mm-hmm. Okay, in the same data set, there's something, there's people close to you who actually, one of these guys responded to a treatment with drug X. And that's a kind of the same situation. And all right, so it could be a likelihood. It's not, and then it's not talking about biology. It's more about maybe random chance, but it's also a different approach to use the data set.
Mm-hmm. Okay. Okay.
Excellent. Just to talk that Philippe Battaglini needed to go out, but he will be with us the next week.
Okay. Well, listen, Marcos. Is there anything else to cover before we end this show?
I think that it's all today. I think that from a part, I think that it's okay, John. We can end it.
Okay.
And I would like to ask to Philippe and Louise if they have something further information to talk to us before we end.
Okay.
No, I just appreciate the invitation again, Marcos and John. It was nice talking to you guys, and with Philippe, it's a long time I don't see him. It's a big pleasure. It's always a pleasure, and talking about personalized medicine is as exciting as it can be. Thank you.
Mm-hmm. Okay, well, this is one of the tools we can use, guys, to really spread education and information and communicate with each other, with people we haven't seen for a while. And after we end this, we'll just kind of chat about what we want to do next week. But this seems to be a really growing field, which will need more education. And this is a good tool, I think, to talk about and to spread the education. So, Felipe, do you have anything else to say, Felipe Ades? Whoop, there he goes. Okay, I guess we'll end this broadcast right now. And hold on, you guys. Hold on. Okay, good day, world.